Dear colleagues, I am honored that I was asked by our dean to introduce you one of our distinguished guests, Professor Zoltán Ivics. He was born here in Budapest in 1964 and went to a second Rákóczi-Ferenc gymnasium here in Buda, and then chose a university a bit east from Budapest in Gödöllő and got a master's degree there in 1988. That is the University of Agricultural Sciences. He joined a research lab interested in animal genetic engineering and started his graduate studies there. And in 1991, he had the chance to visit the laboratory of Perry Hackett at the University of Minnesota, who was a world expert of animal genetic engineering. Finally graduated, got his PhD in 1994 at the Gödöllő University. Stayed in the Hackett lab as a postdoc for 3 more years. And in 1997, he returned to Europe. And after a second postdoc period at Amsterdam, he could establish his first and his own research group in Berlin at the Max Delbrück Center for Molecular Medicine. That was in 1999, and 2011, his lab moved to the Paul Ehrlich Institute, which is in Langen, which is in Hessen, and he still lives in Berlin, so he commutes a lot between the two cities. And there at the Paul Ehrlich Institute, currently he is the head of a research center at the Division of Hematology, Gene and Cell Therapy. Zoltán's major scientific achievement is the development, or I would say the molecular reconstruction of a fairy-tale genetic transposable element called, named by him and his wife, Zsuzsanna Izsvák, Sleeping Beauty. Sleeping Beauty is the first functional gene ever reconstructed from an inactive ancient genetic material. And the Sleeping Beauty-based technology practically revolutionized genome engineering in vertebrates, including human beings. And one of the proof of this that in 2009, this molecule was selected as the molecule of the year. About Zoltán's publication record is more than 160 peer-reviewed articles, which were cited independently more than 15,000 times, according to the Google Scholar. His age index is 67 currently. In 2013, he received the doctoral degree of the Hungarian Academy of Sciences, and 2 years ago, in 2022, he was elected as the external member of the Hungarian Academy of Sciences. He is editor on the editorial board of many prestigious scientific journals, member of the board of the European Society of Gene and Cell Therapy, and in 2018 he was elected as a member of the Academia Europaea. Zoltán supervised 10 10 graduate students, PhD students, inventor of 12 patents related to this transposon technology. He is involved heavily in teaching at the Goethe University Frankfurt, which is near to Langen, since 2011. A few years ago, and also he's a guest professor at the University of Debrecen. A few years ago, I invited him to give lectures at our biology program and also the biotechnology master program, and he happily accepted, and since then he's also with us in the teaching semester. He organized many conferences. I'm just mentioning the last one, which was here in Budapest, the Spring School of European Society of Gene and Cell Therapy, where more than 115 international graduate students attended. Today, Zoltán is going to talk about this fairy tale story. The title of his talk will be Digging in the Dirt, How Archaeogenetics and Synthetic Biology Impacted Gene Therapy. So please, Zoltán, stage is yours.
Egy pár szót azért mondok magyarul is. Először is nagyon szépen köszönöm nekem. Ez egy nagyon nagy megtiszteltetés, hogy itt tudok lenni. Ez az elismerés az egyetemtől nyilvánvaló egy hatalmas dolog az én életemben és a karrieremben. Szeretném megköszönni a karnak, az egyetemnek, a dékán úrnak személyesen is, amit Nyitrai professzor úr nem említett, hogy hogyan is kerültem ide, legalábbis az oktatási tevékenységbe. A biokémiai társaságnak voltunk egy konferencián Pécset, ahol egy asztalnál ültünk, együtt vacsoráztunk, együtt borozgattunk, és egyszer csak elkezdődött közöttünk egy beszélgetés arra vonatkozóan, hogy esetleg milyen potenciális oktatási tevékenységgel tudnék itt hozzájárulni. És akkor ez kialakult közöttünk, és remélem, hogy még hosszú éveken keresztül fog folytatódni. Then I will now switch to English. Also, we have a guest here from Germany for his sake, and also a little bit out of selfishness. I think it's easier for me to talk about the scientific part in English language rather than in Hungarian. So, as Professor Nyitrai already introduced, what I will try to do is to summarize in roughly half an hour what this transposon means, what is the practical relevance of this Sleeping Beauty gene delivery tool. Before I go into the details, let me just introduce to you jumping genes or transposons. As the name already tells you, a jumping gene is an unusual piece of DNA or an unusual gene that can change its location in the genome. Its location is not fixed. It can move about, and just to highlight how this looks like, so here's a transposable element sitting in the genome, just as an example, on a donor chromosome in the nucleus. And what transposition really means is that this transposon will hop over from the donor chromosome onto the recipient chromosome. The two important functional elements, so to speak, that you have to remember out of this presentation is this black piece or black arrow here on the left-hand side, on the right-hand side. These are pieces of DNA that are important for being recognized by a protein which is called the transposase, these orange color circles. And once the transposase protein and this piece of DNA or the transposon DNA are kind of find each other in the genome, then a biochemical reaction will take place that will result in cutting out this genetic element out of this particular position in the genome, and then resulting in moving over or translocating or transposing this piece of DNA onto the recipient chromosome. You cannot get and sequence any genome on this planet without bumping into transposons. This slide, practically speaking, shows to you relative contribution of transposable element DNA in different genomes. Just concentrate on human genome. Roughly half of our own DNA is transposon-based. Roughly 50%, half of this DNA is transposon DNA. What this means, and that's the most important message out of this, if this DNA would all be mobile, hopping, jumping DNA, then we wouldn't sit here today, because this would fragment the genome into small pieces, and then this is not compatible with life. That means transposons are highly regulated to suppress their activities. This is a common theme whenever we look into transposon activity. One man's trash is another man's treasure. What does this mean? What this means is that we can dig in the dirt, as the title suggested, kind of get and isolate these highly regulated, damaged copies of transposons out of natural sources, and maybe we can convert this DNA into a jumping element that also gives us the possibility of using this jumping element as a molecular genetic engineering tool. These were the species, these fish species, where we began to look for these ancient transposable elements that are not active in this fish at all. Very similar situation what I described to you already. These transposons long died out in these fish species over 20 million years ago. But nevertheless, we were able to amplify, by using polymerase chain reaction, the particular gene out of this transposon that encodes the transposase enzyme. Remember, this is the fundamental protein factor that is required for mobilizing these transposons. And when we determine the sequence of these transposase genes, what one recognizes very, very quickly that this so-called open reading frame, basically the coding region of this gene, is full of inactivating mutations. And this is something that I highlighted with these little yellow triangles and other symbols. What this means, in practical terms, you sequence this gene, you bump into these mutations, and you know that this gene cannot encode a functional transposase protein. Nonetheless, what we argued and what we used as a very simple argument is that one can determine a so-called consensus sequence out of this mini gene library. What a consensus really means is that for every single position in this gene, one determines, based on frequencies of occurrences of nucleotides on the level of DNA, or at amino acids on the level of protein, and kind of determine or argue that whatever is dictated by most of this gene must reflect an active amino acid position or active nucleotide in that sequence, because that particular element or that particular nucleotide or amino acid is represented by the majority of these genes. And that's exactly what we did at that time. Today, the way we would do it on a computer screen, generate this consensus sequence, kind of paste that sequence into a small window on a computer, click a button, this sequence would be sent off to a company. Within 2 weeks, I would have the gene cloned into a plasmid vector, kind of a DNA vector, ready to go into an experiment. Now, this is mid-90s. This synthetic DNA technology was not available at that time, so we had to fix all of these little accumulating mutations in the gene one by one by using polymerase chain reaction. And that's what we did, and that's what led to this transposon, which is called Sleeping Beauty. By using the argument, it's a little bit like in the fairy tale. It's a sleeping ancient gene that is kind of inactively sleeping in the genome, is not active. It cannot hop any longer because of these mutations. But now we kind of reconstructed this gene and made this gene active once again in the laboratory. This is just kind of an original sequence alignment that I photographed from the lab. It's kind of a computer-generated printout. Also handwritten notes in there that we used and kind of highlight all the different mutations in the DNA sequence of these genes, just to show you kind of a real-life example how it actually looked like. And many of you may know this movie from Steven Spielberg. It's a science fiction movie, of course. It's how to reconstruct an entire species, an extinct animal, a dinosaur, from ancient DNA. I think we did something similar, except that we didn't reconstruct an entire life form, so to speak, a species, but just a single gene. But out of concept, it's a very similar approach. We took ancient DNA, we fixed the mutations in that DNA, and made that kind of jumping genotransposon alive once again. Now, the practical aspects. Now we have this jumping element that is active in cells. How can we use this for genetically manipulating cells with this as a research tool? So first of all, we can maintain these pieces of DNA I already showed to you on a plasmid vector, on a small circular DNA vector, and practically speaking, incorporate or clone any piece of gene or any piece of genetic information between them. With this, we generated kind of an artificial transposon. This never exists out there in nature. It only exists in the laboratory. Yeah? But this transposon cannot move only if we now take the Sleeping Beauty transposase, we express the transposase transiently in cells. We can supply the transposase in form of a purified protein or messenger RNA or also as an expression vector. And then the rest of the reaction is exactly the same what I showed to you already on the first slide. Namely, this artificial transposon will be cut out from our plasmid vector and will be integrated into the genome of that target cell. In very simple terms, we can take a gene construct, an artificial gene construct or recombinant DNA, and we can integrate this into the nucleus or the genome of a cell at very, very high efficiency. And that opened up new ways of genetic engineering based on this particular transposon. And that's the next area of my presentation. How do we use this Sleeping Beauty transposon for the purposes of curing genetic diseases in humans, what is called gene therapy or therapeutic cell engineering? My first example for this leads me to the hematopoietic system. Hematopoietic stem cells reside in the bone marrow, and they are responsible for generating every single blood cell type in our circulation. Now, if a baby is born with a genetic problem, a genetic disease, a mutation in a given gene, gene therapy in its simplest form attempts to kind of reconstitute the missing function of that gene by moving in a healthy or good copy of that gene into the same genome. So if there's a defective gene somewhere there, the cell doesn't work properly, but now we're taking a new construct containing a healthy functional copy of that gene and we simply stick that into the genome, then we can simply cure that particular cell. And if we do that in a hematopoietic stem cell that we isolate from the bone marrow of this patient, we move in a gene construct, a therapeutically relevant gene construct into that stem cell, and then we put these cells back into the same patient, then these hematopoietic stem cells will give rise through differentiation in vivo in the body to all of the blood cell types that we normally have in our blood system. And by definition, since this gene is now sitting in the genome, every single cell that is differentiated from the genetically engineered blood stem cell will also contain that particular gene that we moved in the first place. This is hematopoietic stem cell gene therapy. Now, here's one experiment. I'll try to kind of formulate this relatively simply. We took a Sleeping Beauty transposon. In this particular case, we just put in a fluorescent reporter gene into the Sleeping Beauty transposon. We mix this together with messenger RNA that encodes the Sleeping Beauty transposase, and then electroporated or moved these nucleic acid components into hematopoietic stem cells. Now, this is a mouse experiment, okay? So it's a preclinical mouse experiment. And then we looked for how efficiently this fluorescent marker gene would be expressed in the resulting genes. And this is the result in vitro. So if you have sleeping beauty transposase present, you see roughly 60% of all of these cells express the fluorescent reporter. If you don't have transposase, and that's the blue line here, this expression is lost. Again, very clearly showing to you that genomic integration of the gene is important. It's a requirement for long-term expression of that gene in these hematopoietic stem cells. And of course, what needs to be done is now to put these stem cells back into the mouse and then see if these genetically manipulated stem cells can actually sit down in the bone marrow and can give rise to cells in vivo. And this is what's shown here. By using the fluorescence reporter in the peripheral blood, in bone marrow, as well as in the spleen, we actually do detect, after 28 weeks following transplantation, the expression of this gene. And what we can also do is now take bone marrow cells from these mice and can put these cells into another animal, which we call a secondary recipient, and also show long-term expression of this reporter system in the mouse. That shows to us that we can take this very simple genetic engineering tool that is based on just two components, a DNA and an honorary component that encodes a protein. We can put these into cells and we can long-term manipulate a hematopoietic stem cell that can engraft and is transplantable in vivo into an animal, into a recipient animal, where it will sit down in the bone marrow, generate cells of the blood system and express that particular gene that we wanted to move in. So what's the therapeutic potential of this? And just want to highlight this particular disease, which is called Fanconi anemia. Fanconi anemia is a genetic disease. It affects any of roughly 29 genes that are involved in DNA repair mechanisms. It is associated with anemia, of course, bone marrow failure, clinically speaking, and cancer predisposition. On a cellular level, Fanconi anemia, since this is a DNA repair defect on the cell level, leads to a sensitivity to so-called interstrand cross-linking agents, for example, mitomycin C. These agents cause trouble in the DNA, and since the repair mechanisms are not efficient, they cannot resolve these issues in the DNA. And very importantly, a proof of concept has been generated by using lentiviral vectors. This is a clinical application where patients are actually treated with genetically corrected hematopoietic stem cells, correcting for the deficiency of the Fanconi anemia mutation. So this is something that we did now, again, in a preclinical mouse experiment. This is what we do, very similar to the fluorescent marker experiment that I showed to you. The Sleeping Beauty construct is also very similar, except that now it has a Fanconi A therapeutic gene cloned into this transposon. We also move these constructs into stem cells in the mouse and then do serial transplantation experiments just to show that fixing these cells can occur with Sleeping Beauty vector. Again, very importantly, when we look at GFP, again, fluorescent marker gene expression over time, we do see an increase in the peripheral blood, these green fluorescent cells. This also shows to you the green cells are the ones that also express, of course, the therapeutic gene, the Fanconi anemia gene. This also means that those cells that are now corrected genetically gain a certain fitness with respect to time. They can propagate better in vivo, and slowly but steadily they take over in the blood system. And these cells can also be moved into secondary recipients and also show very, very decent level, over 60% levels of engraftment of those cells that had been corrected with the Fanconi therapeutic gene. Another area of clinical applications that are very popular these days leads me to CAR T-cell therapy. What is a CAR? It's not an auto. It's an abbreviation of chimeric antigen receptor. A chimeric antigen receptor is an artificial protein that consists of two functional main domains. One, recognition or antibody domain that is responsible for recognizing an antigen that is expressed on a cancer cell, on the surface of a cancer cell. And once this recognition takes place, the cell, typically a T lymphocyte, that actually expresses the CAR would undergo a signaling cascade that gives an instruction to these so-called CAR T cells, or T lymphocytes that express the CARs, to kill the cancer cell. So these are kind of artificially generated molecules that we can incorporate stably in a T lymphocyte, and once the T lymphocyte bumps into a cancer cell, it would kill it. This is how CAR T-cell therapy works. Here on the left-hand side is Carl June, a scientist in Pennsylvania, kind of the godfather of CAR T-cell therapy, clinically speaking. And on the right-hand side is Emma Whitehead, who was a little girl, 6 or 7 years old, when she had CD19 leukemia, and she exhausted every single possible treatment option in the hospital. She would have died. Carl June included her, Emma, in a clinical application with CAR T-cells. This is still kind of early days of CAR T-cell development. And she became very, very famous because she was kind of one of the first examples of highlighting the potential, the fantastic potential in these CAR T-cell therapies. This is Emma Whitehead 10 years later, cancer-free. So that kind of very potentially highlighted the powerful aspects of this therapy. In the European Union, there are 6 products that are currently available for CAR T-cell therapy. 4 of them against an antigen that I already mentioned. This is called CD19, that is expressed on the surface of B cells in the blood system, and 2 of them against the antigen, which is called BCMA. Now comes a little bit of practical considerations. The projection for gene and cell therapy for the next couple of years is an almost exponential increase with respect to the numbers of clinical trials, with respect to the products that will be approved at some point on the market, and of course with respect to market value. This particular slide shows the kind of the projected market value in billion euro in the next couple of years. This really means that manufacturing, and that's kind of the main, at least to me, the main message is that manufacturing efficient, safe, and economically competitive gene therapy products is key to meet the increasing demands in the future. And this is now really important. CAR T-cell therapies in particular, but gene therapy products in general, are associated with very significant prices. And what this, at some point, a health insurance would need to cover the cost of these products. So if a child has a genetic defect like Fanconi anemia, I just use that particular example, or someone develops leukemia, cancer, and then these products would be actually available, so a doctor can prescribe these to you because these are available products on the market. But at some point, someone would need to pay for this. And this is a potential problem, or a very significant problem. Just concentrate on the right-hand side there, which is kind of the price of the CAR T-cell products that are available for the 6 products that I mentioned. So this is anywhere between $300,000 and maybe $300,000 and $500,000 for one shot, just one treatment. And what this results in, and this is very, very important, that in many countries, these are all European Union member states, in some of the countries, these products are just not available because the health insurance wouldn't cover the costs. Just look at Hungary. right here, one particular product, which is Kymriah, that's the very first CAR T-cell therapy or product that became available, is reimbursed by the health insurance system. The others are not. And some other countries are even less lucky. Look at Estonia. If you happen to be Estonia and happen to have cancer, you don't have access to these highly innovative new therapies because this is not recovered by the health insurance. Why is this so? And this is really important now. Because right now, CAR T-cell therapy is undergoing a very sophisticated logistic development. Typically ex vivo autologous cell therapy, which really means that a patient would come into a hospital. The blood cells, typically the T lymphocytes, would be recovered from this patient. This would be transported to a centralized manufacturing site where gene delivery or gene transfer would take place. So the CAR gene would be incorporated into these T cells. Now this product, as soon as it becomes available in this facility, would be shipped back to the hospital that is actually treating the patient. And this logistic circle is very, very expensive and labor intensive. One particular area where our Sleeping Beauty innovation can actually contribute to reducing the cost of CAR T-cell therapy is obviously gene delivery into the cells, because now we're not using viral gene delivery vectors that are actually used in these 6 products that I showed to you, but just use straight nucleic acid-based technology. This is how it works. So now we have a CD19 CAR gene incorporated into a Sleeping Beauty vector. The transposase is also expressed from a separate vector system. We introduce these 2 components into now human T lymphocytes, CD8 T lymphocytes. This is very efficient. Over 50–60% of these T lymphocytes are now CAR positive. This means this chimeric antigen receptor is now expressed on the surface of this T lymphocyte. And now you can take these CAR T cells and then cure a sick mouse with it. This is a xenographed animal model or experiment in which we allow human CD19 positive lymphoma cells to grow. We can visualize these lymphoma cells by using luciferase expression in an instrument. And then a week later, we introduce these human CAR T cells into these animals, and we do show a full elimination of the human cancer cells out of these animals. This is something that CAR T cells can do very efficiently, and now using Sleeping Beauty vector also more economically. This was the very first clinical application in Europe, where I was fortunate to be a member of this particular consortium. It was called Caramba, a multi-center clinical trial that was running in 4 different countries, in Würzburg, in Germany, Italy, France, and then Spain. And just to highlight how this was used, so T lymphocytes were used to kind of transfect the Sleeping Beauty components, and equal fraction of CD4 and CD8 positive cells were mixed together and introduced into the patients. And this is something that is important. The projected cost of just the vector components that we're using for this therapy. You see that the actual investment that you have to put down on the table for these GMP grade vector components is actually quite substantial, but you can treat many, many, many more patients with that same batch of vectors as compared to a classical retrovirus-based vector system in gene therapy. These are just a couple of clinical trials that are running worldwide, mainly in America, using Sleeping Beauty engineering T cells and also B cells, just to highlight that this is now a little bit in the circulation, this particular technology, and there are a number of clinical centers trying to take advantage of this technology. This leads me to summarize this presentation. I started off by introducing to you transposons, or jumping genes. These are plasmid-based, non-viral delivery vectors with a unique capacity of stable genomic integration. As far as I know, a transposon is the only naked nucleic acid-based DNA vector that actually does gene integration very efficiently. Don't forget, transposons have been evolving for millions and millions and millions of years just to do that in nature. And now we capitalize on this particular biochemical reaction for therapeutic cell engineering. Sleeping Beauty transposon vectors allow efficient, stable engineering of therapeutically relevant cell types. And I showed to you examples for hematopoietic stem cells and how these genetically engineered cells can engraft, at least preclinically in animal models. Sleeping Beauty is in clinical testing with genetically engineered lymphocytes. Caramba was one of the applications that has been running here in Europe, but there are a number of other clinical trials that are currently ongoing in America and in other countries. And finally, GMP manufacturing of non-viral vectors has a competitive economic advantage over viruses. And that makes us confident that perhaps at some point, by using this technology, we can lower the price of these CAR T-cell products so that countries would have easier to reimburse the cost of these therapies, so that access for larger numbers of patients would be greater by a contribution of this particular technology. Finally, this is my lab in Langen at the Paul-Ehrlich Institute in Germany. Also wanted to kind of very, very briefly just to highlight some of the collaborators in different countries, including Germany, with thoughts and experimental ideas, and also wanted to also acknowledge a little bit the funding sources that have been going into this particular research. Thank you very much for your kind attention.